sorted see-through here if you don't have a chair. There's one next to me. Speaking of which, there should be various sign-in sheets running through. If you keep passing them along, that would be good, so that everyone who's here can actually sign in and get credit. Hi, well, I'd like to welcome you officially to the first Chichester Colloquium series talk. We're very fortunate today to have our own Dr. Znosko giving the inaugural talk, so I'm very excited for this. Dr. Znosko comes from us from Chichester University. That's where he did his undergraduate research. From there, he went on to Pitt, where he got his PhD in developmental biology. The research he's talking about today, and sort of the exciting thing about this series, is that the people who talk here are talking about the research they're doing on campus in this building. So we've got a biologist today. The rest of the semester, we have a couple of chemists. If you're aspiring to go into research of some sort, this is probably a good place to hear about what the people in your department are actually doing. So, without much further ado, I'd like to introduce Dr. Znosko and his talk, Insights from Zebrafish, a Part Development is Loopy. So, please welcome.
Thank you for that great introduction. I want to thank the committee for letting me do this first talk, and Dr. Fink for kind of starting this colloquium series, so hopefully it will prosper in months to come, years to come. And today I'm going to talk to you about the research that I did for my PhD, looking at heart development in zebrafish. And I'll also tell you about what types of research that I want to start here at Longwood now that I'm here. So if you look at this front picture here, these are the stars of the show. It's a male and female zebrafish. If you look, a lot of times people will ask me, how do you tell the difference between the two? The female is in the foreground, and you can see her large extended belly that's full of eggs. The male is in the background, and he's a lot thinner. So the first question you can ask is, why do I use zebrafish? With so many other different model systems and organisms out there, why is my focus on zebrafish? Well, if you look up here in the upper left-hand portion of this slide, it's just going over the developmental cycle of a zebrafish. And as you can see, at about 2 days after fertilization, this fish embryo looks a lot like a fish. Okay, so you can see proper head structures, proper trunk and tail have already formed, and they're free swimming by this point in time. So just 2 days after fertilization, they're free swimming. It takes about 3 months for them to reach adulthood. Most of the studies I do in developmental biology are within these first 2-day period. Okay? So it's a very quick and rapid life cycle for these organisms. They're easy to maintain, and when the female does lay eggs, she usually lays very large clutches, at least 100. So I have a lot of embryos to work with. Up here on the upper right, I'll just show you a time-lapse video of zebrafish development. And as you can see, if you look on the bottom, this large round circle is actually the yolk of the embryo. And these two balls on top of the embryo are actually the first 2 cells in development. So when you're watching the video, you'll see these cells begin to divide and accumulate at the top. And then eventually there will be a lot of cell movement and migration, and these cells will move down completely around the yolk. And by the end of the video, you'll be able to see different parts of the embryo. So up at the top, you'll see the headed form, you'll see the tail down at the bottom, and then you'll also still see this central yolk. So let's go ahead and get that going. So there's a lot of cell division taking place at the top of the yolk. Eventually, after there's 1,000 cells on the top, the cells will begin to move around the yolk. And then finally, you can see a head with an eye form up here, and there was the tail region down at the bottom. Another reason why we use zebrafish to study developmental questions is that they are transparent embryos. So from the time of fertilization, you can see through these organisms which allows us to ask some pretty interesting and neat developmental questions. So in this example here, here's another video that I'm going to show you different images and how we can use these transparent embryos to study things such as blood in zebrafish. So this is a simple dissection microscope that we're using to look for zebrafish. If we zoom in a little further, you can see the actual red blood cells moving through the brain region. Here's the beating heart at the bottom. Here's the eye of the fish. If we zoom in on another region, the trunk of the embryo, you can actually see the red blood cells moving throughout the trunk, going out in one direction towards the posterior tail, coming back in. If we zoom in even further, we can see the individual red blood cells moving through the smaller vessels within the trunk. Okay, so for all these reasons, and also because there's many different genetic tools for forward and also reverse genetic screens, this was a great model system for me to use to look at things such as heart development. But my research and my lab is primarily interested in developmental biology. So there's a fundamental question in developmental biology, and that basic question is how can you go from one single cell through several different patterning and differentiation and morphogenetic events to finally, at the end, form some multicellular organism. Okay. In my case, it's the zebrafish. So how do we go from an 8-cell organism to form, at 24 hours, several different organs that you can already see, a free-swimming organism, and to an adult form after about 3 months? One of the ways that this is done is through different signal transduction pathways within a developing embryo. The main signal transduction pathway that I focus my research on is called the FGF signaling pathway, or fibroblast growth factor pathway. So just to give you an idea of what is involved in this pathway, you see this phospholipid bilayer here. This is the cell membrane. So this is outside of the cell. This is all inside of the cell. And this is the nucleus of the cell here. So different FGF ligands will bind to cell membrane receptors, and then that will initiate a large signal cascade within the embryo, or sorry, excuse me, within the cell. So it will activate several different proteins, and then once this one protein called ERK is activated or phosphorylated, it can enter the nucleus and then activate different transcription factors. Okay. Transcription factors are important to regulate gene expression. So that's just a brief overview of what the FGF signaling is, but more importantly, why do we care about FGF signaling? So it's been found through a lot of different research that FGF signaling is very important during development. Just for one example, for one FGF ligand, FGF10, it seems to be crucial for limb development. So these are newly born mouse pups, and on the left here, with the skeletal prep and the actual pup, you can see the proper formation of these forelimbs and hind limbs. If you look at a pup that is mutant for FGF10, you can see that, especially particularly when you look at the skeletal prep, it forms no forelimbs or hind limbs. So with just removing one FGF, FGF10, you completely obliterate a limb formation. A couple other examples, another one in zebrafish, you can see that posterior development is crucial, involving different FGF ligands as well. So if you see here on the top, this top panel is a wild type embryo, and as we look down through these pictures, we're deleting more and more FGF ligands. And you can see when we delete the most FGF ligands that this zebrafish here is pretty much lacking the whole posterior region of its body. So that's another example in development why FGFs are important. A final example, and what we'll talk a little bit more about today, is heart development. So it's been shown in the past that an FGF8 mutant in zebrafish doesn't properly form a heart. So early on in development, the heart actually originates as two different populations of cells, which you can see in this embryo. The mutant has a very diminished population of those cells. And if you let the mutant develop a little longer, you can see, compared to the wild type, the atrium in yellow is very much reduced and compressed, and the ventricle in orange is almost completely gone. Okay, so this is another example of the importance of these FGF signals during development. So my lab looks at the FGF pathway, and in particular, within this pathway, what we look at are these X transcription factors here. So in mice and in humans, there are several different subfamilies of these X transcription factors. What I focus on is the PEA3 X transcription factor family, which I have outlined here in green. Now, I don't want you to focus too much on the makeup of these, but I want you to notice that there's two different family members within this subfamily. Again, these transcription factors are important to regulate gene expression downstream of FGF signaling. So before I started my graduate work at Pitt, my boss wanted to do a large screen and look for different expression patterns of different proteins. Okay? So this is kind of just giving you an outline of what we did, but what's important to know is we're looking for expression patterns that are specific to different regions of the fish embryo. So if there's no expression of something in an embryo, we're not concerned about that. If there's expression globally everywhere in the embryo, that's not very interesting to us either. What we're interested in is specific regions in the embryo that are expressing these genes. Okay? So we did a large-scale screen looking for this type of expression pattern. And I'm just going to give you an idea of the kind of results we're looking for. So it is already known the expression pattern of these different FGF ligands. And here I'm showing you different in situ hybridizations during development. And basically what an in situ hybridization is it's showing you specifically where the gene is being expressed. Okay? It's giving you an RNA reading of where that is being expressed. So for example, this is early in development, about 1,000 cell stage, and it progresses through to this is later in development. You can actually see the head of the zebrafish and the eye of the zebrafish here, and the trunk and the tail is off of this picture. It would be down here. So you can see that both of these FGFs are pretty similar throughout development in their expression pattern. Later in development, you can see some expression in the head region. With our large-scale screen, we found a gene, ETV5, that also has similar expression patterns to these different FGFs. So what that's saying to us is, it seems like this ETV5 might be important within the FGF signaling pathway. If it has the same expression pattern as the ligands, maybe it's important in that pathway. So we looked a little bit closer at ETV5. And once we looked at the sequence and did different alignments, and here's just a phylogenetic tree showing you the different relationships, we determined that ETV5 is actually a third family member of this X transcription factor family. Okay? A lot of times zebrafish will have additional family members that mouse or humans won't have because there was a genome duplication event in zebrafish that didn't happen in mouse or humans. Okay, so this ETV5 was a third family member of this X transcription factor family. And we can see that if we look at the expression pattern of these three family members, that they are all very similar expression-wise. So this is about an 18-hour embryo. So again, there's this big yolk in the center, but up here is the head region, here is the trunk region coming around, and here is the tail region at the end. And so all three of these family members have similar expression, and at that same developmental time point, they overlap with the expression patterns of different FGF8 and FGF10 ligands. So we can conclude from this that these family members might be important in FGF signaling. But what their exact role in development and how they're regulated was still unknown. So the questions I have asked in my research before, and I'm continuing to ask, are these three different questions. So I look a lot at the post-translational regulation of these X factors. I'm very interested in the developmental role that these X factors play. And I'm also interested in how these factors bind to target genes. What I feel will generate the most interest with this talk and with student involvement is what kind of roles they play in developmental processes. So that's what I'll talk about. So when I think about what roles these factors play in developmental processes, and if they are important in FGF signaling, then I should look at different developmental processes that involves FGF signaling. Some of those processes are brain formation, early heart development, and late heart development. And with zebrafish, we have a great tool, a great technique that we can use to study the X transcription factors, or any other different types of transcription factors. And this technique is called Morpholinos. Okay. So I had to throw some structure in there for the chemistry people in the audience. But what Morpholinos do is they can bind to the initiation complex of a particular mRNA. So you design these Morpholinos for a particular sequence, and they'll bind to the initiation complex of the mRNA of that sequence. So it creates some type of steric hindrance where it prevents the ribosome from binding to the mRNA. And we all know if the ribosome can't bind to the RNA, then you can't make any protein from that. So basically what we can do is we can knock down the protein levels of these transcription factors during development and see what happens if we eliminate them from the equation. What will happen to the development of the embryo? So for example, if we look at the FGF signaling pathway again, if I knock down these X transcription factors, then they are important in FGF signaling, we would think that all these downstream targets, these genes that are regulated by these transcription factors, will also get knocked down. So we can inject one-cell stage embryos with these Morpholinos, let the embryos develop a little bit, and then look at the expression levels of downstream targets. In addition to that, I can also look at developmental processes that are important in FGF signaling, such as brain development or heart development. So to first, just to give you an idea of the kind of results that I'm looking for, here I'm looking at two different downstream targets of FGF signaling. And across the top here are a bunch of different Morpholinos I generated. Okay, the control Morpholino is just used as a control. It shouldn't affect the development at all. These other three Morpholinos that I injected all target X transcription factors. The ETV5 Morpholino targets ETV5, and the 2X Morpholino targets 2 of the family members, and the 3X Morpholino targets all 3 of those family members. So when we look at the expression of downstream targets, you can see that if you knock down more and more of these family members, so if you look at this last column, you can see the expression of these downstream targets is reduced. You see a lot less purple in these embryos than you do in the control embryos. But if you just look where we only knock down one family member, you don't see too much of a difference. So what we're thinking is going on is they kind of make up and fill in for one another. So if we just knock one family member down, the other 2 family members will pick up the slack. But if we knock down more and more, or all 3 of them, then you really start to see a reduction in the downstream targets. So if we look a little further at different developmental processes that require FGF signaling, one of them was brain development. So if we look a little later at brain formation in the embryos, again, this dark spot here is the eye, and this band here is showing you where a portion of the brain is beginning to develop. Again, if we knock down more and more of these transcription factors, you see that the brain development is pretty highly reduced. Which brings me to the most interesting part of my talk, I feel, and that's the formation of the zebrafish heart. Okay, so the zebrafish heart is very similar to human hearts, other mammal hearts, mouse hearts. So what we learn with the zebrafish heart, we can really relate to other mammals and humans as well. So I first just wanted to give you an overview of how this zebrafish heart actually forms. And I alluded to this earlier, but the heart kind of first forms as 2 different populations of cells. So you can see these 2 different populations of cells on either side of the embryo here. Okay. Eventually, as development takes place, these 2 populations of cells will finally come together, which you see here, and they'll form a single tube. Okay. And that's all considered early heart development. And then, just showing you through these pictures, the tube will begin to extend and become longer. And then finally, what happens is this looping event. So normally the tube is somewhat straight, and then at about 4 days, you have this looping event that kind of puts a kink in the heart. And that's when the heart is finally divided into different chambers. So the zebrafish heart is only a 2-chambered heart. It has 1 atrium and 1 ventricle, but this looping, this little kink here, divides the tube between the atrium and the ventricle. So I wanted to look and see what happens if I knock down these transcription factors, what will happen to heart development. So I looked at both early heart development, at these 2 different populations of cells, and then I looked at later heart development when the heart loops. So if I look at early heart development, here's just a representation of the 2 different populations of cells. And as you can see, it can be subdivided into smaller little populations. And some of these populations of cells will give rise to different things. So for example, these populations of cells down here will give rise to the heart. And these populations of cells in blue up here will give rise to blood and vessels. So I did in situs and looked at the expression of all these different genes and looked at what happened to them when I knocked down these transcription factors. Okay, again, I tested a bunch of different morpholinos, and I'm knocking more and more transcription factors down as you progress to the right-hand side. And as you can see, GATA4 is one of these genes that is expressed in this heart region. And as you can see, as you move to the right, the expression pattern of GATA4 decreases. Okay. The same thing happens with this heart-specific gene, NKX2.5. Same thing happens. As we inject more morpholinos and knock down those transcription factors, we're seeing less and less heart formed. And a third gene that gives rise to heart structures also has the same result. Okay. But I mentioned that up here, this blue region, it's adjacent to the cells that give rise to the heart, but this blue region gives rise to blood and vessels. So what I see when I look at the expression of this gene when I use morpholinos is I actually see an expansion of those cells. Okay, so if you look here at the control, and then you look as I inject more morpholinos, you actually see that amount of cells, or that area, is expanding. So what we think is going on is that these X transcription factors are important in FGF signaling pathway. And what their important job in heart development, at least early on, is to maintain these heart lineages. But if we knock down these transcription factors, then they can't properly maintain these heart lineages. So they get smaller. And because they get smaller, other cells beside it, other cell populations, have room to expand. So what will give rise to blood or vessels, those regions will expand, and the heart regions will get smaller. So that's an idea of what's going on early in heart development. If we look a little bit later in heart development, when the heart actually comes together, those populations come together and form a tube, we also see effects when we inject morpholinos. So the tubes just don't look shaped properly. If you look at these wild type pictures and we're staining for different regions of the heart tube, and then you look at our morpholino injected embryos, they don't look like they're the same shape. But we weren't exactly sure what was going on. So we let the zebrafish develop a little more and looked at the heart after it made its looping event. And then we saw some really interesting results. If you look here under wild type, the heart loops in such a direction that it's called an L-shaped loop. So it looks like the letter L after the looping event is taking place. But what we saw when we injected more morpholinos and knocked these transcription factors down is the looping of the heart actually is reversed. So it doesn't look like an L anymore, it kind of looks like the opposite of an L. And that was a very interesting result for us because we kind of were alluded to some of the importance of these transcription factors in development. Okay. Because we were seeing this mislooping event, that alluded to us that there might be problems in left-right asymmetries within this organism. So what does left-right asymmetries mean? So here I have just examples of different regions of a developing embryo and time progression down the side here. So if you look early on in several different regions of the embryo, everything is pretty much symmetrical from the left and right hand side. So in the guts, in the heart, in the lungs, everything's symmetrical. As the embryo gets older, into 7 weeks or 11 weeks, you can see that these asymmetries start to form. So for example, the liver is on one side of the body and not the other. The heart is positioned to one side. The different lobes of the lungs are different depending on which side of the body you're on. So these different asymmetries seem like they might play a role in heart development because you have a specific looping pattern. And when we knocked down these transcription factors, we saw an alteration in that looping. So the final little bit that I want to talk to you today about is, how does that happen? How does left-right patterning happen in the zebrafish, and what role do these transcription factors play in that process? And this is kind of where the really loopy part of the talk comes into play. So it's been looked at and studied a lot and determined that the different left-right patterning of zebrafish embryos is all due to this little fluid-filled sac at the tip of the tail that's stained in blue in these pictures. Okay, here's just another view looking at the tail, and you can see that small circle fluid-filled sac. And what it's thought to do is it contains a lot of cilia. So if you can see in red there, it's a bunch of different cilia within that sac. And what people think happens is this cilia will beat in a certain direction. And it will move different molecules on one side of the embryo or the other side of the embryo. So just the movement of the cilia and the beating of the cilia will cause certain molecules just to be on one side and not the other. Okay, here's an example of a video of this actually occurring. So people injected little beads into this fluid-filled sac, and you can actually see the beads moving around. So we'll let it play here, and it's zooming into that sac, and you can see the red beads moving around in a circle because of the cilia beating in a certain direction. So we wanted to look at that and see, if I knock down these transcription factors, what happens to this whole process? And we had a great way to study this because we created this transgenic line here. And I'll show you the development of this transgenic line in a second, but it's expressing this GFP, this fluorescent protein, and it's a downstream target of FGF signaling. So it'll be expressed in all the regions that are FGF active. And one of those regions is this fluid-filled sac at the tail. So I can see what will happen to this expression if I inject my morpholinos and I knock down these transcription factors. So that's what I tested. I tested in this transgenic line what would happen if I injected these morpholinos. And you can see in the uninjected control, you can see a large amount of GFP expression in that sac. But after I injected the embryos, you see the GFP expression go way down. And that's kind of what we expected. This is a downstream target of FGF signaling. If these transcription factors are involved in FGF signaling, it should knock down that expression. But if we just looked at the overall formation of this sac between injected and non-injected, it looks fine. It forms fine. So we figured, well, if it forms fine, it probably functions fine too. But what we wanted to look at next was the actual cilia inside of that. So when we look at the cilia, then we saw a dramatic difference. So if you look at the cilia and the control and count the number, you see about 50 cilia within this sac here. But when we look at Morpholino injected embryos, that number is diminished by over half. So it has half as many cilia there. And remember, I said cilia are important to move molecules on one side of the embryo or the other. So if there's not enough cilia, maybe that won't move the molecules to one side. Maybe that's not enough force to move the molecules. And so we looked at that. So this is expression of a gene that's only on one side of the embryo, only on the left side in wild type. When we injected our morpholinos and looked at expression, which all three of these are morpholino injected, you can see the expression is completely randomized. You see some that have no expression at all, others have expression just on the right-hand side, some have expression on both sides, so it's completely randomized. Kind of the conclusion, what we think is going on is in wild-type embryos, you have this fluid-filled sac, and the beating cilia is important to move certain molecules on one side of the embryo. And with that, you get proper cardiac looping. When we knock down these transcription factors, we reduce the amounts of cilia in that sac. And so it can't properly beat and move expression to just one side. So sometimes it can't move expression at all, and you'll have a mislooped heart. Or other times there's enough cilia to move some of the molecule on one side, but not all of it. So under those circumstances, you'll just have a straight heart, not looped to one side and not looped to the other side either. So that was kind of the conclusion to our studies. So just very briefly in the next couple minutes, I just want to tell you how I want to incorporate this in my lab here at Longwood. So like I mentioned before, there was this large-scale screen done to look for any different genes that have kind of neat expression patterns. And so we have a bucket load of genes with pretty cool expression patterns, and we have no idea what they do. So that's one of the things I want to do. So here's just some examples. Obviously, these haven't been categorized because we just named them with random numbers and letters. Okay, but they're in interesting areas. They're in the brain region. Some are at the very tip of the tail where this fluid-filled sac is. So this is something we really want to look at further. Okay, and some of them have similar expression patterns to FGS. So you can ask a slew of questions of what we can do to try and look at the function of these unknown genes. The same thing for other genes that are just expressed in that fluid-filled sac as well. So I have a slew of those too, and we can ask the same type of questions. What happens if we knock these down? What happens if we overexpress them? What if we do the opposite? What roles do they play in development? And that's one of the main things that I want to look at. One of the other main things that I want to do, and if you're more interested in things like ecology, I want to test and see what different local environmental pollutants, what kind of roles they play on the development of zebrafish. Okay, so right here in our backyard, what kind of effects are these pollutants having? And people do these studies very commonly with zebrafish, and this is just an example of a paper I found that's doing that. And even more interesting is they are looking a lot at heart development in this process, and they found a lot of different pollutants will affect the development of the heart. So that's another thing that I'm really interested in. If you're interested in things like environmental factors, that's another project that I'm looking to spearhead too. Okay. So with that, just want to conclude. Hopefully you got some of these ideas from my talk that FGF signaling is important and crucial for development. that this set of transcription factors is important in specific developmental processes like brain formation and early and late heart development, and that these factors are important in maintaining the number of cilia in this fluid-filled sac as well, and that has a role in left-right asymmetry. So a lot, and most of that work I've done was from Pitts, and these are all the people that helped me there, my advisor and other lab mates. And just want to thank you all for coming. If you have any questions at all, I can try and answer them for you. And that's it. Thank you. Yeah.
When you added, like, you off those two, was it always the same two, or did you try, like, see if different combinations of these?
Right, so
Could you repeat the question, please?
So the question was, so there's 3 members in that family of proteins that I looked at. So when I knocked them down with Morpholino, she's asking, did I knock down different combinations of those 3? Trying to get an idea of maybe is one more important than the other 2, or is one essential and the other 2 are just kind of filler? And so yes, we did do that. And it appears that there's no hierarchy within them. They all appear to be important. If you knock out any 2, you'll see a good effect. If you knock out all 3, you'll see a good effect. And no matter the combination, you'll see similar effects. Yeah.
So you got this little spear in the tail developing embryo, cilia inside it. They're beating a certain direction. Fluid flow determines left-right symmetry.
Right.
My question is that the universal determinant of left-right symmetry? Meaning, if you could get the cilia to beat all in the opposite direction, would you end up with an adult zebrafish that is the mirror image of a normal zebrafish?
Right. So it's not known in zebrafish if it is the only determinant, but it appears to be a very important determinant. So if you, for example, people have done studies where cilia beat because there's a little motor in every cilia. And so people have done studies where they've kind of stopped that motor from working. So there's cilia there, they're just not moving. And they see a lot of left-right defects. No one has done any way of kind of reversing the beating of the cilia to see if they can see a mirror image. But it looks like just altering these cilia alone without doing anything else has an effect. So we can't say yet that it's the only thing, but it's definitely important enough that without it, you don't get proper left-right. Yeah?
How difficult was the quantification of the amount of Morpholino that you will need in order to completely shut down the mRNA? Or is there some leaking going on?
Okay, so we did a lot of work with that. I didn't go into that on my talk. But it's important to emphasize that we can't say that it's a complete knockout. Okay, there's maternally contributed amounts of these signals that we can't knock out. But anything that's made after fertilization, and once transcription and translation begin, we can knock out. So we call it a knockdown because there's still a little bit left in there. What I didn't tell you about the different concentrations I used is I used a ton of concentrated Morpholino when I would just knock down one family member, and I would never see an effect. When I came to knocking down 2 or all 3 family members, I had to greatly reduce the concentration, and I still see large phenotypes. Okay. If I use the same concentration in all 3 as I did in the 1, you completely kill them. So it's very dose dependent.
Thank you.
Yeah.
Doc, is there any corollary to this FGF signaling in humans or other animals? If you take this model and apply it?
Yeah, so pretty much across the board, FGF functions in similar ways in mice, in humans. So FGF has been shown to be important in heart brain and limb development. So the three biggies for FGF across the board.
Okay, so I like the limbs. Is the one. FGFs interact pretty specifically with sonic hedgehog and homomorphogenetic protein. If you don't have FGFs and sonic hedgehog and DSPs working together, you don't think the right way.
Right.
So if you're knocking down FGF here, are your fins deformed as well? Do you not pay attention to them if you're so interested in art?
Right. So FGF is important for fin formation.
Right.
Yeah. We can look at fin formation with our specific transcription factors because fin formation, the earliest you can look is 48 or 52 hours. And that's pretty much the latest we would ever look at heart development. So we never carried it out to look at fin formation, but FGFs do play a role in fin formation. I just don't know about my specific transcription
Will they
factors.
stand for you?
Yeah, they can live longer. I was just more interested in other things. Yeah, but it can be something we can easily look at. Thank you. Thanks for coming.